Hi guys, welcome back to my channel, and today we're going to learn about DNA replication. So before going into the topic, we should know about the central dogma of molecular biology. That is, the flow of information from DNA to RNA to protein is termed as the central dogma of molecular biology. So whenever DNA will replicate itself, the process is called as DNA replication. Likewise, when there is synthesis of RNA from DNA, the process is called transcription. And whenever there is synthesis of protein from mRNA, that is process is called as translation. And today we're going to learn about DNA replication. The DNA replication is a process in which DNA copies itself to produce identical daughter molecules in order to transfer of genetic information from parent to their progeny. Now, we should know this DNA replication is the semi-conservative method because this DNA is a double-stranded structure, and in the process of DNA replication, the one strand will be the original strand and another will be the new strand. So this is known as the semi-conservative method. In this process of DNA replication, we should know about a lot of enzymes and a lot of steps. So first of all, we should know where is the origin of replication will start from. So a quick revision of the DNA, we have already discussed about it in our previous videos, but let's learn a quick review. This DNA is a double-stranded structure and it is anti-parallel to each other. That is, one will run in the direction of 5' to 3' and another strand will run in the direction of 3' to 5', and they are complementary to each other. That means adenine will make bond with thymine and guanine will make bond with cytosine. This AT will have 2 hydrogen bonds and the CZ will have 3 hydrogen bonds. So what we can know from here, in the DNA where there is more AT joints or bonding, there will be easy to unwind this DNA because it have only 2 hydrogen bonds and it is comparatively unstable as compared to that of the CC bonds. So now we should know how this DNA get unwind. So this is the double-stranded structure of DNA, and now it should be unwind, and this process will be done by the enzyme called DNA helicases. This DNA helicase will unbind this DNA, and there will be another very specialized structure that is known as SSPP. That means single-stranded DNA binding protein. So what this does is it keeps the strand separated and provide protection from nucleases enzymes also. So there are two main important things that we have to know is that DNA helicase unbinds this DNA, and this SSPP, that is the single-stranded DNA binding protein, will help to keep this DNA strand separated, and this DNA helicases uses the energy in this process also. Now we can see there is formation of something like this Y-shaped structure. This is the Y-shaped structure. Now this particular thing is known as the replication fork. Now the unwinding and separation of these 2 strands of DNA results in the formation of replication fork, and it moves along the DNA as the synthesis progresses. Now we have to know about the enzyme DNA topoisomerase. There are 2 types, one is the type 1 and another is the type 2 topoisomerase. So in the process of this DNA replication, there may be supercoiling of this DNA. So to prevent this supercoiling, there should be one enzyme, and that is the DNA topoisomerase. It also uses energy and helps in the to prevent the supercoiling of the DNA. Now there is another enzyme known as DNA polymerase. There are two types of DNA polymerase. One is I and another is III. We will briefly know about this DNA polymerase 1 lately. Now let's focus on this one, DNA polymerase 3. So it have two main functions. One is it will synthesize DNA and another is the proofreading part. We will discuss about it later. Now this DNA polymerase will synthesize DNA, but it cannot initiate the synthesis of DNA. It requires another thing that which is known as the RNA primer. So in the process of DNA, there should always be this RNA primer which is produced by the enzyme primase. Only that DNA polymerase can initiate the synthesis of DNA. Now this DNA polymerase is something called the builder part, but the very important thing that we have to know is this only builds strands in 5' to 3' direction. This RNA polymerase will make DNA in 5' to 3' direction only. Now this is our DNA. So this is the replication fork. This is our DNA. This DNA have two strands and one strand is known as the leading strand and another is known as this lagging strand. And we know this DNA polymerase will make DNA in the direction 5' to 3' only. So, okay, I'll make it in different colors so that we can understand it. So if there is DNA polymerase here, it will make DNA in the direction 5' to 3'. So this part of this process will be the continuous process. There will be the continuous synthesis of DNA and this is the leading strand. But in opposite, if there is DNA polymerase here, it will make from 5' to 3'. But as the DNA is unwinding, okay, I'll make it clear here. So here the 5' to 3' structure is here and it is going in this process in this continuous, but here now 3' to 5' structure is made, but as the DNA progresses there will be the gap here. So again it should make another process from 5' to 3'. So this is not continuous, this is the discontinuous synthesis. So there will be fragments of DNA and now this fragments of DNA is known as the Okazaki fragments. So these Okazaki fragments will be bind to each other, that will be linked to each other by another enzyme which is known as DNA ligase. You can easily remember by this L, this ligase will link. It acts as the glue that will join this Okazaki fragments. Now, this DNA polymerase 3 have two functions. One is it will synthesize DNA and another one is it proofread the newly synthesized DNA. That is, it ensures that there are no any errors in the replication. So it checks if there is nucleotide sequence correctly placed or not. And very Interesting thing is we have already discussed that is DNA polymerase always synthesize DNA in the direction 5 prime to 3 prime, but it can proofread in 3 prime to 5 prime direction also. That is, while synthesizing this DNA in 5 prime to 3 prime structure, it can also come back in 3 prime to 5 prime structure in order to check if there is any error or not. So it also has this exonuclease activity. Now let's learn about this DNA polymerase 1. This DNA polymerase 1 will remove this RNA primer. So it has the activity of 5 prime to 3 prime exonuclease activity. That means, first of all, there was RNA, that is the RNA primer, so that DNA polymerase can act on that. But we don't need that RNA because we are in the process of DNA replication. We don't need any RNA here. So this RNA should be removed, and the removal of this RNA primer will be done. This function will be done by this DNA polymerase 1. So this is all for DNA replication. It might be quite confusing, but it is easy. So let's quickly summarize the process of DNA replication. So first of all, there is DNA, which is a double-stranded structure. We are going to replicate it, so we should unwind the structure. For unwinding, there is the enzyme known as DNA helicases, and to support this, there is single-stranded DNA binding protein, which will keep this DNA structure separated. And there is the enzyme called DNA polymerase 3 that will synthesize DNA with the help of RNA primer, which is produced by the enzyme called primase. And whenever there is RNA primase, this DNA polymerase 3 will start to make DNA in the direction 3 prime to 5 prime to 3 prime. And in the leading strand, it is the continuous process, but in the lagging strand, it is not continuous process, it is the discontinuous process. So these fragments are called the Okazaki fragments. These are linked up or joined by our DNA ligase enzyme. And to prevent the supercoiling of our DNA, there is the enzyme topoisomerase, which will prevent the supercoiling of DNA. Likewise, to remove this RNA primer, there is another enzyme, that is the DNA polymerase 1, that will remove the RNA primer. And DNA polymerase 3 have another function of proofreading also. So this is all for DNA replication. We will learn about transcription and translation in our upcoming videos. So thank you. Hope you guys enjoyed, and we will meet up in our next video.